Hello, in this video, I'm going to talk about muscle protein adaptations to aerobic endurance training. So I want to start by clarifying how research is done on muscle adaptations to aerobic endurance exercise. So most of the studies are looking at what happens in the case of chronic muscle stimulation. So where they actually apply electrical stimulation to muscles for at least 8 hours at a time, and sometimes up to 24 hours a day. So it's not the same thing as whole body endurance training, but it's meant to simulate what's happening in muscle tissue during endurance training. So it's not exactly the same. But it does produce results that are happening in the same direction as when endurance training is studied in humans with actual voluntary exercise as opposed to chronic electrical stimulation. So the results are usually more extreme in chronic muscle stim studies than they are in voluntary exercise training. But the results are still happening in the same direction. So generally, you're still seeing the same results, just not as extreme because the methods aren't as extreme. So I just want to highlight a few key differences between chronic electrical stimulation and whole body endurance training. So one, I already mentioned that in chronic electrical stim, the duration of the procedure is usually between 8 and 24 hours a day, and then that's usually ongoing for however many weeks or depending on the protocol. The chronic electrical stim studies often don't include rest, which could make a difference in how the muscle proteins are adapting or what's being up or down regulated and so on. So it could make a difference. And in chronic electrical stimulation, it causes the same pattern of activation to be imposed on all of the motor units where the electrical stim is being applied, which is different from voluntary movement because we know that there's an enormous amount of variability in motor unit recruitment, the firing frequency, patterns of firing, and changes that happen with fatigue. And so those things are sort of overridden when we use chronic electrical stimulation. And then lastly, chronic electrical stimulation doesn't involve voluntary recruitment, as I just discussed, but that also means that the contribution and response of the brain and spinal cord to the movement is going to be different than it is in voluntary movement, where the brain and spinal cord play a pivotal role in control and recruitment. Okay, so with all that said, everything I'm going to share with you here is based on studies that have, some of them are chronic electrical stimulation studies, but that are also backed up by in vitro studies, animal studies, and other methodologies, not just in chronic electrical stim. So everything here is supported and is not only true in the case of electric stim. So muscle protein adaptation. So muscle tissue adapts to stresses imposed by regular exercise. So depending on the type of stress, we have different adaptations to be able to cope with those stresses. So depending on the type of stress, we might have upregulation or downregulation of different specific proteins. So we'll start to synthesize more of a certain type of protein or less of a certain type of protein, or different isotopes or different types of proteins, depending on the type of stimulus or the type of exercise that's stimulating those changes. So those changes, so more or less of different types of proteins, those alter the properties of the muscle fibers to allow those fibers to respond better when faced with that same type of stimulus. So if, like, let's say it's endurance training that we're exposing the fibers to, the types of proteins that are being produced in the fiber are going to change in favor of improving performance and fatigue resistance and damage, and improving those things, those different characteristics of the muscle fiber, so that next time we go through the same type of endurance exercise, that we're better prepared to cope with that, and that the muscle is able to prevent damage, perform better, and avoid fatigue for a longer time. So the best strategy for a muscle fiber to adapt its proteins is to adapt all of the proteins together, as opposed to like this protein and then that type of protein and then that type of protein. Ideally, we want to adapt all of the proteins in a sort of synchronized, coordinated way. Otherwise, you end up with some proteins that are adapted, but that need to work together with other proteins. And if those haven't also adapted or been changed to a different type of protein, then it may not be able to withstand the specific needs or the specific type of demands that the new proteins are putting on the old proteins. So ideally, we want all of those proteins to adapt in a coordinated way so that the muscle is able to act the most efficiently. We're not wasting proteins or destroying any tissue or anything like that because we have the wrong types of proteins that are paired. So I think I just skipped a slide here. There we go. Pre-translational control is referring to the control of protein synthesis by alteration of the abundance of mRNA. Okay, so this is before we are producing the new proteins. This is how we're stimulating and controlling which types of proteins that we're producing. So first, mRNA stands for messenger RNA, and the mRNA is essentially like photocopies of the DNA. So DNA is what is holding on to our cellular instructions, to our genetic code. And the DNA is sort of like the original copy. And we don't want to just spread the original copy around the cell. We want to keep it safe so that it doesn't get damaged or start to send the wrong signals to different parts of the cell. So we keep the DNA safely stored inside the nucleus of the cell, but the other parts of the cell need instructions that come from the DNA so that they know what types of proteins to make and how big and so on. So the mRNA, those are basically like commands that are carried from the DNA, like copying certain instructions from the DNA and carrying them to other parts of the cell to deliver those instructions so the other parts of the cells know how to build the right proteins. So there's different kinds of mRNAs that promote synthesis of different kinds of proteins. So pre-translational control is essentially where we're putting out more or less of different types of mRNA to stimulate more or less production of those particular types of proteins. So concentrations of different mRNAs change in response to each bout of exercise. So when we exercise, we are up and down regulating different types of mRNAs so that we start producing more or less of the different types of proteins, depending on the type of exercise that we just performed. So this is happening while we are actually exercising, but it seems like the most significant change in the concentrations of the different mRNAs are probably happening during the recovery after exercise. Okay, so it starts happening during exercise, but when exactly it happens is different for the different mRNAs for the different proteins. So it seems like mostly they're happening during recovery, but some are happening more so during exercise, depending on the type. So how the concentrations of the different mRNAs change will depend on the intensity of the stimulus. So how intensely are you working? So is it moderate intensity, cardio exercise, vigorous? Is it strength training? What is the stimulus and how intense is it? And then this process is also going to depend on other things like nutrient levels, hormones, temperature fluctuations, and all sorts of other variables. So for the most part, this is how it works. But if there isn't enough protein and there's not enough amino acids to construct the protein, or there are hormones that are sending opposing signals or that sort of thing, then that could interfere with this process or enhance this process, just depending. Okay, translational control. So this is when we're actually synthesizing the new proteins. So translation, the process by which a protein is synthesized from the information in the mRNA. So translational control means that we are changing the amount of protein synthesized per unit of mRNA. So in the last step, we talked about how we'll have more or less of different kinds of mRNA to stimulate synthesis of different types of protein. But even though we're up and down regulating the mRNA, we also can control through various mechanisms how much protein we synthesize in response to each unit of mRNA. So translation can also be altered to coordinate the expression of two proteins whose functions are closely linked but have differing transcription rates. Okay, so what we're saying here is that the goal is coordination. Remember, it's more efficient to have turnover of multiple proteins at once, especially the ones that are going to work together within the muscle fiber. So we want to coordinate the translation so that we have expression of multiple proteins at the same time. And so that's what we're saying here, is that during the translation, we are manipulating that to control how many of what we're producing. Okay, now after those proteins have been synthesized, then we have post-translational modifications that could occur. So that includes things like incorporating the protein that was just produced into the muscle fiber, transporting the protein within the cell, so moving it from one place to another, and breaking down of existing proteins. So again, we want to coordinate the replacement of old proteins with new proteins, and we want to coordinate this happening at the same time, at least within a functional unit within the muscle fiber, as much as that's possible. So that includes breaking down old proteins, weighting, transporting, incorporating proteins into the fibers. So just because a protein has been synthesized during the last step doesn't necessarily mean that it has been kind of built into the muscle fiber and the protein complexes in the muscle fiber. So that's happening during post-translational modifications. We can modify what we do with that protein, how quickly, and again, we can work there to coordinate the replacement of those proteins. All right, so signals to trigger this adaptive response that I just described. It's not totally clear exactly how the muscle knows to go through all of these processes and to start replacing proteins and to replace with different types or what kinds. So it's not totally clear how that signal is happening, but there are 4 methods of signaling that are most likely, and they are probably happening all together. So it's probably some combination of these 4 methods of signaling. So there's metabolite signaling. So there are changes in concentrations of different metabolites that are caused by repetitive muscle contraction. So there's changes in all sorts of things inside of the muscle fiber that happens in response to repetitive contraction, like we see in endurance training, where we have ongoing repetitive muscle contractions to cause repetitive movements. And so some of the things that might be triggering this adaptive response could be the level of ATP within the cells, the level of AMPK in the cells, and the ratio of ATP to free ADP. So those are just a few things, and there are other metabolites that also could be at play here. And then calcium signaling, kind of similar to metabolite signaling, but changes in calcium level or concentration of calcium in the cell during increased contractile activity could also be signaling the cell to make these adaptive protein changes. And then mechanical signaling. So there's something called MAPK pathways. So MAPK signaling is when we have a chain of proteins in a cell that send a signal from a receptor on the surface of the cell to the DNA that's contained in the nucleus. So it's the way that the signal is transmitted from the outside of the cell to the nucleus of the cell. So it's possible that MAPK pathways are activated when the stress of exercise is imposed on the cell, and that causes that signal essentially to be sent via the MAPK pathways to the nucleus to cause these changes in the cell. So mechanical effects, that could be like metabolic changes, calcium changes, mechanical stress on the cell itself, and those could potentially all activate the MAPK pathways. And then lastly, are hormones. So hormones and autocrine or paracrine factors. So hormones are chemical messengers that we secrete, and those go through the blood to act on other cells and organs and things. Autocrine is when a cell secretes a hormone and that hormone acts on that same cell that secreted it. Paracrine is when a cell secretes a hormone and it acts on the neighboring cells. So the surrounding cells nearby, the one that secreted it, that'd be paracrine. So during exercise, there is stimulation for secretion of different hormones, whether autocrine, paracrine, or otherwise. And so that includes insulin-like growth factor, vascular, endothelial growth factor, just as a couple examples. But insulin-like growth factor is responsible for growth and development of tissues. So it makes sense that we would secrete insulin-like growth factor in response to exercise. And vascular endothelial growth factor is a chemical that triggers angiogenesis, so genesis or creation of new blood vessels. So the more we are exercising a muscle, the more blood it requires to sustain that activity, because it'll get more oxygen and nutrients and so on from the blood. And so more activity means we need more blood supply to that muscle. So the muscle cells will actually secrete vascular endothelial growth factor to signal growth of more blood vessels in that area. All right, thank you so much for watching, and I'll see you in the next video.